Hello everyone, welcome to SRIRAM IS. I welcome you all to the platform of Prelims through PYQs, where we are discussing different set of questions. Today we'll be discussing the Biotechnology 2. We have already made a discussion for the Biotechnology 1. In today's session, we'll be looking into the important questions that was asked in the Biotechnology, right? Now, for all the questions that I'll be discussing, I'll be giving you some insights and what are the most likely part that can be again reiterated in the terms of questions in the civil services examination. For the first question, this is from 2020. We already had the questions starting from 2024, right? And we completed in 2020. There's certain number of questions that is still left out. Biotechnology being again very important and significant portion in science and tech. So we'll see the important questions. In this, there are certain statement which is given. The first statement is the genetic change can be introduced in the cell that has produced egg or sperm of a respective parent, right? Now see, when it comes to the all genetic changes, and mitochondria is the one that is taking most of it part. So if you look at this particular statement, the genetic changes can be introduced. Yes, it can be introduced. Is it possible with part of certain technology? Now the second is a person genome can be edited before the birth at the early embryonic stages. Now this is again feasible and practical if you look at the part of technology which is there, even in vitro fertilization, in vivo fertilization, this is something most likely to happen and this can take place. Now the third is human induced pluripotent stem cell can be injected in an embryo of a pig. Yeah, this is something again possible. When you see the nature of the pluripotent cell, it can definitely be, the part of its potential, it can be done, and this can be injected into the embryo of pig. So considering all the three statements that is correct, the correct answer here is D. Now what more you need to focus on this particular question, specifically when it requires you to understand about the mitochondrial change that can be done specifically in the parents, and categorically in mother, it is important for you to know that the changes and the attributes coming from the mitochondria is mostly from the mother. The other you need to understand is about the embryonic stages, right? And the third is about the pluripotent. So do have a read of all these things so that if any time the question appears on these same topics, you are easily able to answer that. Now coming to the second question, that is what is the importance of using pneumococcal conjugate vaccine in India? Now, if you look at the part in 2020, this was again in the news, government has been making its proactive efforts with regards to pneumococcal vaccines. Now, the statement which is given is these vaccines are effective against the pneumonia as well as meningitis and sepsis. Now, this is true because if you look at the streptococcus disease which is there, was mainly to curb the part of this. So pneumococcal conjugated vaccine was there only to ensure that the streptococcus virus which is there, the bacteria which is there, has to be curbed out. The second is the dependence on the antibiotics that are not effective against the drug resistance bacteria can be reduced. Now this is again something very true on this particular part. The third, this vaccine have no side effect and cause allergic reaction. So it says that it has no side effect and it causes no allergic reaction, which is not true. See, any vaccines which is there specifically for conjugate vaccine, it has certain part of repercussions in the body. It can have allergic reactions. There are other part of the body functions might have certain miscalculabilities which can be there. So in this particular part, this is not true. So the correct option here is 1 and 2, which is option B. So make sure some basic parts can confuse you about all these things. Some generic term might be confusing. So if you have a clarity on that, if you use a basic understanding and basic common sense, you will be easily able to answer that. Moving ahead with the other question, this was asked in again 2020. We can see a good number of questions coming from 2020. See, each year the dominance of biotechnology in the science and tech paper is there. Now, be it the common general science or the science and tech portion, apart from the tech, biotechnology will always have a substantial contribution. So in context of the recent advantage of the human reproductive technology, what is pro-nuclear transferase? Now this recent is in that context of year 2020. Now we are more focusing on pro-nuclear transfer. What is this pro-nuclear transfer? See, I'll show you a diagram to make this understand in a better way. So pronuclear means, see, this is the pronuclear transfer in human embryo. What actually happens, some mRNA, the mitochondria which is there from a parent couple. You can see this image, it will help you understand. So this is where the part of the fertilized egg without a faulty mitochondria, the part of mitochondria is being removed, and the donor couple, this is where how it is being. Now this, you can see this images where the patient nuclear DNA is transported into the egg with a healthy mitochondria. This is where the replacement takes place, and after 6 to 8 days the old embryo or mitochondrial disease get removed. Now, to answer this question, it was asking about the pronuclear transfers where the mitochondria is being transferred. So the correct option here in this particular question is, no, you have already talked about, this is not the correct answer, that the fertilization of egg in vitro by the donor sperm, genetic modification of sperm producing cells, right, development of stem cells into the functions embryos, right, and prevention of mitochondrial disease in the offspring. So this is the correct option. Now moving ahead with the other question. RNA interference, or better known as RNAi technology, has gained popularity in the few years. This was specifically in the year 2019. So make sure you have a check on the years as well. So RNAi is actually known as post-transcriptional gene silencing, right? Now slicing is again a part which has been used many a times in various other questions. If you look at the question of CAPF and CDS, UPSC has been asking questions, right? So be rest assured that if it is being asked in RNAi or post-transcriptional gene silencing, you need to be aware of both this stuff. Now what are the options that is given? It is used in developing the gene silencing therapy. Absolutely correct, because it actually, the purpose of this RNAi is to ensure that the gene silencing and certain therapies to ensure that certain diseases that is coming the way has to be stopped. Now the second is, it can be used in developing therapies for the treatment of cancer. Yes, absolutely, whatever the part of deletion can be done through this technology. Now the third is, it can be used to develop hormone replacement therapy. No, it has nothing to do with the hormone replacement therapy, which is again a different domain of understanding. Now the last is the option is, it can be used to produce crop plant and resistance to various pathogens. So only 3 is not the correct option. Apart from that, 1, 3 and 4. If you look at the part 1, 2 and 4 is the correct option. The correct option here is A. Now moving ahead, what is Cas9 protein? That is often mentioned in the news. This was again asked in 2019. Cas is some of the important technology which is also called as CRISPR. I'll show you through the images to help you understand. So CRISPR or Cas, these are what is being used. This is called clustered regulated interspace short palindromic repeats. You can see the repeats which is going. This is where the targeted part is actually taking place, and there are certain seizures that is being used, right? So this is the image that will help you understand how the seizures take place, and certain part of the cell is being replaced, right? Now what is the question that is being asked? It is asking that how this protein is actually related and what it is doing, what is the purpose of it. So it's a very basic and generic question. It is asking, molecular seizures is used in targeting the gene editing. Absolutely correct. This is the one. A biosensor used in the accurate deletion of the pathogen in the patient. No, this is not the correct one. Then it is asking that a gene that makes plants pest resistant. See, it has nothing to do with the part of resistance. It is actually a molecular seizures. This Cas protein is an enzyme, right, which is helping in identifying the specific location. Now the last but not the least, it is asking that it is a herbital substance synthesized in a genetically modified crop, which is absolutely incorrect, right? So the correct option here, it's a molecular seizure used in targeted gene editing. Now I have already talked about this, so make sure you know the complete full form of CRISPR or Cas9, clustered regulated interspace short palindromic repeats. Now moving ahead with other questions with reference to agriculture in India, how the technique of genome sequencing often seen in the news can be used with immediate future. Now, genome sequencing has been a favorite topic in civil services examination. UPSC has been asking a lot of questions with regards to gene edit and gene sequencing. Now, the questions that is there is genome sequencing can be used to identify genetic markers for disease resistance and drought tolerance for the crop. Absolutely correct. If you look at the part of purposes which is specifically there for the part of genome sequencing is to identify and rectify the error which is there, right, irrespective of plant. Now, second is this technique helps in reductions of the time required to develop a new variety of crop plant. Absolutely correct, because we'll be ultimately saving the time. Third, it can be used to decipher the host pathogen related in the crop. So all the three options that is given here is correct. See what happens if you know the basics of this genome sequencing, why it is used, what is the purpose, what is the significance, what are the advantages, how it is actually helping in the farm sector or how it is actually helping the specific crop. If you're able to comprehend that, you will be easily able to answer these particular questions. Now, the other question is, what is the application of somatic cell nuclear transferase technology? Now, this is again something very important. This is called as SCNTT, right? This was asked in 2017. So, it is asking what is the application where it can be used. Productions of bioclavirids, manufacturing of biodegradable plastics, reproductive cloning of animal and productions of organism for free diseases. Now, I'll show you an image which will give a more clarity to understand what exactly this technology is all about. So, you can look at this image. This is somatic body cell with desired genes. So, what happened if this is nuclear fused with denucleated egg cell, right? You can see these are the egg has been denucleated and this is the egg cell of the nuclear removed. Now, once this is being fused and the clone cells are produced, right? So, this is where the surrogate mother, or if you look at the therapeutic part, the cloning culture can take place. So, it can be used either in the reproductive culture or it can be used in a therapeutic cleaning. Now, to answer your questions, what application it has been used, it is actually used for the productions of reproductive cloning of an animal. See, if you understand the basic term, the statement which is given become easy for you to comprehend, right? So, make sure you are able to relate it with the concepts. Now, moving ahead with other questions, in the context of the development of bioinformatics, the term transcriptome, sometimes seen in the news, is related to what this was asked in 2016. Now, to help you understand what is transcriptose and other terms which can be important in the examination, I'll just deal each one by one. The first is genome. What is genome? Where the coding of the genes are actually being discussed or been studied. Second, it is for the non-coding genes. Now, transcriptome is actually the mRNA. What is mRNA? mRNA is nothing but it's a messenger RNA, right? It has a small and non-coding RNAs and long non-coding RNAs. Now, then we have proteome. What is this? This is a structural protein. This is a non-structural protein, functional protein and enzymes. Then the last we have metabole. What is this? These are the lipids, amino acids, carbohydrates and nucleotides. So, you can make out for the transcriptome, it is actually the mRNA. Let's see the options which is given. It is a range of enzymes used in the genome editing. It has a full range of mRNA molecule expressed by the organism. Third, the descriptions of the mechanism of the gene expression, absolutely not. And mechanism of genetic mutations taking place in the cell. The correct option here is B, that the full range of mRNA molecule expressed by an organism. Right? So, make sure if you are reading, try to relate the other terms which is associated with it, because there might be some questions coming out of that specific term. Now, moving ahead with the other questions, that is the Genetic Engineering Appraisal Committee, we commonly call it GEAC, right? GEAC, Genetic Engineering Appraisal Committee, was constituted under the options that is given is Food Safety and Standard Act. See, this was an act which was actually there for the Food Safety Authority of India, Food Safety Authority of India, right? We have geographical indications of goods. This is Registration for Product Act 1999. This is for the GI tagging. This is for GI tags. Then we have Environment Protection Act. This is 1986 and Wildlife Protection Act 1972. The correct option under which the Genetic Engineering Appraisal Committee was formed was the Environment Protection Act of 1986. Now, what is important from the Genetic Engineering Appraisal Committee, that they might ask you about the functions, and even they can ask you about the nodal ministry which is working. So, Ministry of Environment Forest, MOEF, is a nodal ministry which is actually taking care for the entire functioning. UPSC in its civil services examination might ask you about the basic functioning of it, right? So you need to check what are the functions, how this particular body is very important to give an assent in conducting the part of genome, the things which is there is given by the Genetic Engineering Appraisal Committee, and even it gives certain part of restriction. If you look at the Dhara, the GM mustard which was approved by it, Supreme Court has still not given the full-fledged approval to proceed for it. So just understand the power and functions of the ministry. This might be important for the upcoming exams. And this was asked in year 2015. Now moving ahead with the other question, consider the following technique and phenomena, right? What are the technique and phenomena? This is budding and grafting of a fruit plant. This is the one. Second is cryptoplasmic and mere fertility. And the third is gene silencing. Now, if you look at budding and grafting in a fruit plant, this is nothing short of technique, which is usually a basic phenomenon which is taking place for the fruits and plant. Grafting is something you must have studied in NCERT for class 9, 10th, right? Now, coming to the cryptoplasmic and plant fertility, now this is one of the technique involved to ensure that any part of the malfunctioning which is there in the genes can be improved. The same goes for the gene silencing. If the things is not working, if you want to delete something, this part of gene silencing can help in deletion and rectification of the further improved version. So for this, the correct option, absolutely 1 is not the one. We have the correct option, which is 2 and 3. So this was all about the questions that we have discussed till 2015. The rest question which is still there from the biotechnology, human disease, will be discussed further in the PYQ discussion. I would like to highlight that do ensure that you are putting your impetus and focuses on the biotechnology portion. It is substantial portion in the tech which will have you and give you some marks which is close to 7 to 10 marks, right? So this is something which you should not take a leverage of, and definitely try to ensure that you are putting all the best possible effort apart from all the static subject that you're reading. Science and tech, I have always been vocal about this, that this is an easiest topic to score the marks. If you know the term, if you know the basic statement, basic definition of it, you can easily able to answer it. So I wish you all the best for your preparation. We'll meet in the next session. Thank you.